Welcome to Eduimagica, the medical mystery education hub. Today we are going to learn about beta-oxidation of fatty acids. And in this video we'll cover up all important points related to it. Beta-oxidation is a catabolic pathway of fats. In the beta-oxidation of fatty acids, free fatty acids are converted into acetyl-CoA, and during this conversion, oxygen is utilized in one form or the another. That is why this process is also known as oxidation. Now, there is a very important point. Now the question arises why this process is known as beta oxidation. So to understand this, we have two images on screen which depict the structure of fatty acid. As we all know that the fatty acids are composed of long carbon chain and a functional group attached to it, that is the carboxyl group. And we also know that the carbon atom that is directly attached to the functional group is known as the alpha carbon, and the carbon atom next to it is known as the beta carbon. And in the case of the beta oxidation, the bond between the alpha and the beta carbon, or the bond between the alpha and the beta R chain, gets broken down, which results into beta oxidation. Now let's talk about the storage of fats. As we all know that fats in human body are stored in various forms, but the major storage form is triglyceride. And if you talk about the triglyceride is composed of glycerol molecules, and the glycerol molecules is attached to 3 molecules of fatty acids. Now moving forward, let's talk about the overview of the reaction. Human body contains adipose tissue which consists of triglyceride, and triglyceride is composed of 1 molecule of glycerol attached to 3 molecules of fatty acids from our previous knowledge. Now these fatty acids are distributed all over the human body, and from there it enters into the blood circulation, and from blood they enter into the every cell of the human body that can metabolize free fatty acids. Now, this is also a very important point that in the human body there are 2 tissues which cannot metabolize free fatty acid, that is RBC and the nervous tissue. This is so because both of them have one thing in common, that they lack mitochondria, and mitochondria is essential for fatty acid metabolism. Now let's talk about the stages of the beta oxidation. There are 3 stages of the beta oxidation. The stage 1, transport of fatty acid from adipose tissue to target cell. The stage 2, activation and transport of free fatty acid inside the target cell mitochondria. Third stage, oxidation catabolism inside mitochondrial matrix. Now we will study about each stage in detail. First of all, the stage 1, that is transport of fatty acid from adipose tissue to target cell. As we all know that fat consists of adipose cell, and adipose cell consists of triglyceride. If you take a look in the adipose cell, triglyceride is composed of glycerol molecule that is attached to 3 molecules of fatty acids, or we can also say that it is attached to 3 molecules of free fatty acids. Now here in this place an enzyme comes into play, that is the pancreatic enzyme, that is lipase. The lipase breaks the bond between the glycerol and the free fatty acids. So after the bond is broken, the free fatty acids moves directly into the blood circulation, and from there it moves into the every cell of the human body that can metabolize free fatty acids. Now suppose here is a cell that can metabolize free fatty acids with the help of mitochondria. Now the free fatty acids are transported into the cytoplasm of the target cell by the help of a special protein, fatty acid transporter. The free fatty acids cannot move into the membrane or the cytoplasm because it has a net negative charge, so they need such proteins for the transport, that is the fatty acid transporter. Now we will talk about the free fatty acid transport into the mitochondria. To understand this, we will first look at the membranes of the mitochondria. The outer membrane is outer mitochondrial membrane. and the inner membrane is inner mitochondrial membrane, and between them we have the intermembrane space, and inside inner mitochondrial membrane we have mitochondrial matrix, and the free fatty acids which need to be transported into the mitochondria need to be activated, because the free fatty acids cannot utilize mitochondria as such. Now in the stage 2, we'll talk about the activation and transport of free fatty acid inside the target cell mitochondria. First, we will talk about the activation of the free fatty acid. In the activation of the free fatty acids, the reaction occurs in 2 steps and requires ATP, coenzyme A, and the magnesium ions. The free fatty acids are activated to acetyl-CoA by the help of an enzyme thiokinases or the acetyl-CoA synthetases. The fatty acid reacts with ATP to form acyl adenylate, which then combines with the coenzyme A to produce acetyl-CoA. 2 high-energy phosphates are utilized since the ATP is converted to pyrophosphate. The enzyme inorganic phosphatase hydrolyzes pyrophosphate to phosphate. The immediate elimination of pyrophosphate makes this reaction totally irreversible. And we all know that fatty acids cannot be utilized in the mitochondrial membrane as such, so it needs to be activated. Now this is very important point. Please pay attention that the acyl and the acetyl groups are totally different. If we talk about the acyl group, in the acetyl group, it is the combination of acetate or the acetic acid, that is 2 carbon unit with coenzyme A, whereas the acetyl-CoA means acyl group, any fatty acids C4 to C26 in length combined with coenzyme A. Now we will talk about the transport of activated fatty acid inside mitochondria. As we all know that mitochondria has 2 membranes, and on the one side we have the cytoplasm, and on the inner side we have the matrix. The outer membrane is known as outer mitochondrial membrane, the inner is known as inner mitochondrial membrane, and in between the outer mitochondrial membrane and the inner mitochondrial membrane we have intermembrane space. And in the outer membrane space, mitochondrial membrane, we have a pore-like structures known as porins, and due to the presence of porins, it becomes permeable for acetyl-CoA. So the acetyl-CoA can easily get into the intermembrane space, but the acetyl-CoA is impermeable to the inner mitochondrial membrane. So for this we need a very special mechanism known as carnitine shuttle mechanism. The carnitine shuttle mechanism contains various enzymes. The very first enzyme is present in the outer mitochondrial membrane, that is carnitine palmitoyl transferase 1. This carnitine palmitoyl transferase 1 attaches one molecule of carnitine to acetyl-CoA and transfer it to the intermembrane space, forming acyl-carnitine. Now this acyl-carnitine is need to be transported into the matrix by the help of an enzyme acyl-carnitine translocase. Now in the inner mitochondrial matrix, we know that acyl-carnitine cannot be utilized as such, so it needs to be converted into acetyl-CoA. So for the conversion of acyl-carnitine into the acetyl-CoA, we have another enzyme, that is carnitine palmitoyl transferase 2. Now what is the role of this enzyme is that this enzyme attaches one molecule of coenzyme A to acyl-carnitine and it forms acyl-CoA, and along with the acyl-CoA it also regenerates one molecule of carnitine, and this regenerated carnitine is transferred back into the intermembrane space by the help of acyl-carnitine translocase. Now we'll talk about the third stage, that is oxidative catabolism inside mitochondrial matrix. Now suppose we have a fatty acid structure. In this we remove OH from the chain and we attach coenzyme A, so it becomes fatty acyl-CoA. And suppose we have a carbon chain length of 16 carbon, so it is known as palmitoyl-CoA. And if we have a carbon chain of 14 carbon, it is known as myristoyl-CoA. And if we have a carbon chain of 12 carbon, it is known as L'Oreal-CoA. And during its conversion, two products are formed. And during the formation of the two products, four enzymes are used in this reaction. So it forms one molecule of acetyl-CoA and new fatty acyl-CoA, which is 2 carbon short in length. In the case of palmitoyl-CoA, it will be myristoyl-CoA. This myristoyl-CoA re-enters until it is completely utilized. Each cycle of beta-oxidation liberates a 2 carbon unit acyl-CoA occurs in a sequence of 4 reactions. That is, first is dehydrogenation, second is hydration, the third is dehydrogenation, and the fourth is cleavage or thiolysis. First we'll talk about the dehydrogenation reaction. In this reaction, we have acyl-CoA, and in this case, we have a 16 carbon length, that is palmitoyl-CoA. So it gets converted into trans-acyl-CoA by the help of an enzyme, that is acyl-CoA dehydrogenase. And in this reaction, the hydrogen is removed from the alpha and the beta carbon and the double bond is formed. And during this reaction, FAD is reduced to FADH2. And in this case, if acyl-CoA dehydrogenase is deficient, it leads to SCID. SIDS. In the second reaction, we have hydration. And in this case, the trans-acyl-CoA, which has a double bond between the alpha and the beta carbon, it gets converted into beta hydroxyacyl-CoA. And in this case, one OH group is attached at the beta carbon and the double bond is removed. And in this case, a molecule of hydrogen, a molecule of water is also used. Now we'll talk about the third reaction, that is the hydrogenation reaction. And in this reaction, beta hydroxyacyl-CoA gets converted into beta keto-acyl-CoA. By the help of an enzyme, beta hydroxyacyl-CoA dehydrogenase. And in this reaction, a molecule of NAD is also reduced to NADH. And in this reaction, at the beta carbon, a double bond O is attached instead of OH. And in the fourth reaction, that is the cleavage reaction, the beta keto-acyl-CoA, that is a 16 carbon in length, gets converted into two products. That is, first it forms acetyl-CoA, and on the other hand, it forms the acyl-CoA, that is 2 carbon shorter in length from the parent chain. That is, in case of the palmitoyl-CoA, it will be myristoyl-CoA, and the myristoyl-CoA will be reutilized in this reaction until and unless it is completely oxidized or utilized. And in this reaction, it will be utilized until and unless 8 molecules of acetyl-CoA is generated. Now let's talk about the yield of the beta oxidation of fatty acids. Now in this, suppose we have a palmitoyl-CoA that is 16 carbon long. The palmitoyl-CoA forms molecule of acetyl-CoA, and during this process it releases or form one molecule of NADH and one molecule of FADH₂. And for the formation of 8 molecules of acetyl-CoA, it needs 7 cycles of beta-oxidation. So to understand this, we must first understand that one molecule of acetyl-CoA is equal to 10 ATPs, one molecule of NADH is equal to 2.5 ATPs, and one molecule of FADH₂ is equal to 1.5 ATPs. So in the 7 cycle of beta oxidation, we have 8 molecules of acetyl-CoA. Along with it, we have 7 molecules of NADH and we have 7 molecules of FADH₂. And now let's talk about the different things. The 8 acetyl-CoA utilized in the Krebs cycle and the 7 NADH is utilized in the electron transport chain along with the 7 FADH₂. So the ATPs found by the 8 acetyl-CoA is 80 ATPs and the ATP found by the 7 NADH is 17.5 ATPs and by the 7 FADH₂ it is 10.5 ATPs. Now if we talk about the total yield or the net ATP yield, it is 80 ATPs plus 17.5 ATPs plus 10.5 ATPs, it will be 108 ATPs. But we will deduct 2 ATPs from it because these 2 ATPs are utilized in the activation of the fatty acids. So net ATP we get is 106 ATPs, that is 106 ATPs per molecule of palmitoyl CoA. That's all for today. Thank you for watching. You can follow us on Instagram, Facebook and Twitter. The links are in description below. You can also visit us at www.eduimagica.com. Do subscribe and hit the bell icon so you don't miss any updates.